Welcome to the HEOR Insights YouTube channel. Today, we're diving into a really fundamental topic, the grade approach. This is all about how we assess the certainty of evidence, which, as you'll see, is absolutely crucial in our field. So, here's our game plan. We'll start with the basics. What exactly is grade? Then we'll get into its certainty framework. After that, we'll walk through the 5 key reasons you might downgrade evidence. And finally, we'll tie it all together and see how grade works in the real world of HEOR. All right, Section 1, what is grade? You see, in health economics and outcomes research, the question isn't just, does this treatment work? That's only half the battle. The real question we need to answer is, how much can we actually trust the evidence that says it works? And that's precisely what grade helps us figure out. So, what does grade actually stand for? It's the grading of recommendations, assessment, development, and evaluation. Now, here's a really important distinction. Grade isn't about looking at one study in isolation. Not at all. It's a framework for assessing the entire body of evidence. That means all the studies we have for one specific outcome. It's about the big picture. At the end of the day, grade helps organizations like health technology assessment bodies answer this one critical question. Is the evidence strong enough to justify the cost? Think about it. Payers need to be confident in the clinical data before they agree to reimburse a new treatment. Grade is what provides that structured way to build confidence. So let's move on to the core of the system, the certainty framework. This is where we get into the nitty-gritty of how grade actually classifies evidence into 4 different levels of confidence. Okay, so there are 4 levels. High certainty is the gold standard. It means we believe our model inputs are really solid. As we go down to moderate and low, that confidence starts to waver. We're introducing more risk into our economic models. And if we land on very low, well, that's a huge red flag. It basically means the evidence just isn't strong enough to make a good decision. Now, this is a super important rule within grade, the starting point. Where your assessment begins depends on the type of evidence you have. If you're looking at a body of evidence from randomized controlled trials, or RCTs, you start at the top, high certainty. But if your evidence is coming from observational studies, you have to start at low certainty. From there, you can adjust that rating based on what you find. All right, Section 3. This is where we really get into the how. We're going to break down the 5 domains, or the 5 main reasons, why you would downgrade the certainty of evidence from its starting point. So, we systematically check our evidence against these 5 areas. First, risk of bias, then inconsistency, indirectness, imprecision, and finally, publication bias. If we spot any serious issues in one of these domains, we have to downgrade our certainty level. First up is risk of bias. This is all about the methodological flaws or limitations within the individual studies themselves. We're essentially asking, were these studies designed and conducted in a way that minimizes the chance of getting the wrong answer? So to do this, we use standard tools, like the Cochrane Risk of Bias tool, for example, to check each study. We look for things like a lack of blinding or high dropout rates. If the overall result we're seeing is mostly coming from studies with these kinds of serious flaws, we have no choice but to downgrade our confidence. Here's a perfect example. Imagine you're looking at quality of life data from open-label trials where everyone knows who's getting the treatment. For a subjective outcome like that, the potential for bias is huge, so you'd absolutely have to downgrade for risk of bias. Next up, domain number 2, inconsistency, which you might also hear called heterogeneity. Here, we're asking if the results from the different studies are telling a similar story. If they're all over the place, it's hard to be confident in the overall conclusion. So what are the warning signs? Well, you might see that the results of the studies are wildly different, or maybe their confidence intervals barely overlap. We also have statistical tools to help us, like the I-squared statistic. If you see an I-squared value over 50%, that's a big sign that the studies are pretty inconsistent with each other. For example, let's say one trial shows a really strong treatment effect, a hazard ratio of 0.60, but another one shows a very weak effect, like 0.95. If you can't find a good reason for that huge difference, you have a problem with inconsistency, and you've got to downgrade. Okay, on to number 3, indirectness. I'd say this is probably the most common reason for downgrading evidence in HEOR. The question here is pretty straightforward. Does the evidence we have on hand directly answer the specific question we're trying to answer for our decision? Indirectness can pop up in a few ways. Maybe the patients in the trial aren't like the real-world patients who will get the drug. Or, a very common one, the trial used a placebo, but what we really need to know is how it compares to the current standard of care. Or maybe the outcome measured isn't the one we truly care about. A classic example of this in HEOR is when you're building an economic model. The model needs overall survival data to work properly, but the clinical trials only reported on a surrogate endpoint like progression-free survival. That's a major mismatch. It forces us to make assumptions, which introduces uncertainty and means we have to downgrade. The fourth domain we look at is imprecision. This one is all about statistics. We're asking, is there enough data here to give us a precise estimate of the effect, or is the result so uncertain that it's hard to draw a firm conclusion? We mainly check for imprecision by looking at the 95% confidence interval. If it's really wide, that's a sign of uncertainty. And a huge red flag is if that interval crosses the line of no effect. For a ratio, that would be 1.0. If it does, we can't even be sure there's any benefit at all. Another clue is just not having enough data. Too few patients or events can lead to an imprecise result. Let's look at an example. Say your overall result is a risk ratio of 0.85, which suggests a 15% risk reduction. That sounds pretty good, but if the confidence interval is from 0.70 all the way to 1.05, that means the true effect could be anything from a 30% benefit to a 5% harm. That's too uncertain. That's imprecision. Time to downgrade. And finally, our fifth domain, publication bias. This is a tricky one. It's the risk that the evidence we can see isn't the whole story, because studies with negative or boring results just never got published. They're sitting in a file drawer somewhere.
How do we spot this? Well, sometimes a graph called a funnel plot can show a lopsided pattern that suggests studies are missing. But often, it's more of a qualitative judgment. If you do a search and all you find are a bunch of small, industry-funded studies that all just happen to be positive, that should make you suspicious. You'd expect some small studies to show no effect just by chance. If they're all gone, that's a sign of a problem.
So if your systematic review on a new supplement finds 10 small studies that all show a benefit, but you can't find a single small study that shows no effect, that's a classic signal of publication bias. The evidence would definitely need to be downgraded. Okay, so we've covered the theory. Now, for our final section, let's connect all these dots and look at how grade ratings actually impact the day-to-day work of HEOR, especially in HTA submissions and economic modeling. Let's walk through a realistic scenario. Imagine a submission where your evidence for overall survival comes from a big, well-run RCT that gets a high certainty rating. But the quality of life data, even from that same trial, gets downgraded to low certainty because of bias and imprecision. What happens? Well, an HTA body like NICE would probably accept your survival data as their main analysis, but they would absolutely challenge that quality of life data, probably asking you to run scenarios assuming it has zero effect. The whole grade assessment process is neatly wrapped up in something called a Summary of Findings table, or an SOF table. This is a standard part of any good systematic review. It's a transparent way to show for every important outcome what the effect was, what the final certainty rating is, and what it all means in plain language. So here's the key takeaway. The next time you see a headline about a study's results, don't just ask, how big was the effect? The more important question is, how certain is the evidence? Because in the world of HEOR, a huge effect size means very little if the evidence behind it is rated very low. Understanding that difference is absolutely essential.